So this is the structure of alpha-ketoglutarate. I'll come back to it in terms of what it does to the cell a little bit later. We decided to test it among about 30 or 40 other natural products reported to affect aging because of this paper in 2014, which showed that it could extend lifespan in worms. And Gordon's lab was able to repeat these findings very robustly, and so we started studying them in mice. And this is a data I'm going to show you has been accepted in Cell Metabolism and will be coming out soon. What we did is initiate treatment with alpha-ketoglutarate at 18 months of age in both male and female mice. And if I just showed you this slide, you might not be that excited because the effects on lifespan are modest, about 10% in females and about 5% in males. It's significant in females. It doesn't quite reach significance in males. We repeated the study again and found almost identical results. If you combine the two studies, which you're not really supposed to do, but I'll tell you anyway, then you reach significance in males. But the effect on lifespan is not what got us the most excited. What really got us excited was the effect on health span. And to do that, we monitored frailty. And so we used this 31-point frailty index in mice that was developed to be relatively non-invasive and to be analogous to, as far as possible, to frailty indices in humans. And this was developed by Susan Howlett in Canada. And so we have started using this frailty index in all of our experiments on aging because we like it so much. So I'll just take you through this slide. On the left, you're looking at each dot as an animal at a different age, 18 months all the way up to 31 months. And this is a frailty score. And you can see that the control animals in blue, the frailty goes up faster than the AKG-treated animals in, I guess, magenta. But this is kind of a misleading statistic because what's happening is animals are dropping out of the statistic because they're dying during this window. And so in other words, if you take this animal here that had a very high frailty score at 23 months, it doesn't show up at 25 months because it died in the interim. And so to show this data more clearly, what we did is we regraphed the data after all the animals died in terms of the percent of the animal's lifespan when we took the measure. So in other words, all of these points occur within 50 and 60% of the lifespan of the animal, 60 and 70%, 70 and 80, etc. And what you can see is in the control animals, frailty goes up pretty steadily over the course of age. But in the AKG animals, it's largely blunted. In fact, what we see is a 50% reduction in frailty in the female animals. I'm not going to show you the males, but we see almost the same effect. It's about a 40% reduction in frailty. And that's led us to conclude that AKG is having the effect of compressing morbidity in these animals. So it's extending lifespan, but it's extending health span and reducing frailty much more dramatically. The animals stay healthy for a very long time, and then they decline very rapidly at the end. Here's another way of presenting the data. And here what we've done is ignored the animals, the longitudinal effect of the study, and just plotted the frailty score versus the remaining lifespan, either for control animals or for AKG-treated animals. And interestingly, if you look at the females and the males, frailty scores in control animals predict the remaining lifespan of the animals. So you can see a very good p-value for both females and males. But if you look at the lines in the magenta, there's almost no slope to those lines. And in fact, the frailty score very minimally predicts frailty in females and doesn't predict frailty in the males. So what that again suggests is that these animals are very protected against frailty, even though they eventually die slightly after the control animals. Just to show you the animals themselves, this is 27 months of age. Control animals are having all kinds of problems. Loss of hair, hair graying, although that's a little bit hard to see. Coat condition is dramatically impaired. You can see other kinds of problems in different animals. The IKG-treated animals are largely protected from especially the frailty score, the frailty components that relate to coat condition. Their coat remains much better during this period of time. They're also more active. And what we've been trying to understand mechanism, and one of the things I'll point out, there's a lot more data in the paper, is that AKG seems to enhance the stem cell function in the skin or in the hair bulbs. So melanocytes are preserved with AKG-fed animals in green, although they're lost by this time point in the control animals, accounting for changes in hair color. We also believe that other adult stem cell populations are likely to be preserved in AKG-treated animals as well. We're looking at this in muscle stem cells and tracheal stem cells at the moment. And this may be one common feature by which AKG protects animals with aging. Now, if you go back to this molecule, this is a very well-known molecule, alpha-ketoglutarate. All of you that have had a biochemistry class have talked about it. It's in the middle of the TCA or citric acid cycle, so in central metabolism. It's important for converting glucose into components that can feed into mitochondrial pathways and generate energy. But I should also point out that alpha-ketoglutarate is involved in a large range of enzymatic reactions in the cell. It's involved in amino acid synthesis and probably is a substrate or cofactor for hundreds of different enzymes in the cell. It's a little bit like NAD, and many of you have thought about NAD in aging, is that this molecule is doing so many different things in the cell that it's a little bit hard to understand which of those might be beneficial for aging. And so we see a lot of benefits when we administer AKG. We see preserved adult stem cell function. We see better metabolism, and I would say that as metabolic flexibility. One reason I say that is AKG is a central component converting amino acid catabolism and carbohydrate metabolism, so it helps cells convert back and forth between different energy sources, and that seems to be preserved with aging. We see enhanced respiration, altered microbiome function. This is something we're studying in great detail, in more detail, and then a dramatic effect on inflammation, and I'll show that to you in a minute. In addition, we're starting to see changes in aging biomarkers, including in humans that are taking the product produced by PDL and reporting their epigenetic clock scores back to us. I'll come back to that later. So there are a lot of different components and pathways of aging that are affected by AKG. This inflammatory component is quite dramatic. These are different inflammatory cytokines here. You can see that from 18 months to 28 months, many of these boxes become more red in the control animals, indicating higher expression of inflammatory factors, and much of that is blunted with the AKG-treated animals. You can integrate the scores of all these different components and come up with a general inflammatory cytokine index and show that that's dramatically reduced by alpha-ketoglutarate treatment. I won't go into too much more detail on that, but this may be one of the central pathways that's being affected by AKG, although we don't yet know how. Now, I want to come back to something that more recently we're studying, and this is data that's not yet going to be published, but we were interested by the fact that alpha-ketoglutarate has been widely used to enhance exercise performance. I just pulled out 3 things off the internet here, showing suggesting increased athletic performance, better muscle recovery from wounds and surgery, and also improved bodybuilding and other components of exercise. And one of the things that people taking AKG have reported to us is they see improved exercise performance. And often AKG is given with arginine, another amino acid. So we're not doing that. We don't necessarily recommend supplementing amino acids. We're just looking at alpha-ketoglutarate by itself. But we wanted to look into this in more detail because the mechanism is not entirely clear for what might be happening. And one thing that jumped out at us, and the reason we got interested in this, I should say, is that in our studies, we were finding that AKG is not really taken up by cells at a very high level in culture. When we give it to animals, we can see that it gets taken up in the intestine, goes in the bloodstream, gets to the liver, but many other cell types don't seem to take it up. But in looking back at the literature, which is always a good idea, my postdoc, Guan Choping, found in 1963 that AKG was reported to be utilized by red blood cells for glutathione synthesis. This is actually before I was born. I'm not quite that old yet. And so we've been looking into this in somewhat more detail. Now, red blood cells don't really have mitochondria, so making glutathione and many other molecules has to occur through different pathways. And this has been studied more as the time has moved on. And so one study, in fact, found that 89% of the glutathione in red blood cells in tracing studies was derived from alpha-ketoglutarate. And this is a mechanism by which glutathione is made. It's basically a combination of 3 different amino acids, and alpha-ketoglutarate is a key component of glutamate synthesis, which is one of those components. So red blood cells take up AKG quite dramatically. If you look at our data, what we found is either in vitro, in cell culture, administering AKG to red blood cells, you can get a pretty dramatic increase in glutathione synthesis, or in vivo, although this is very preliminary data, we can see increased glutathione levels in red blood cells when we administer AKG. We would have a lot more data on this right now, but we've kind of got halted by COVID-19, so we're just getting back to these studies right now to try to confirm this finding. It's a little early. But when you take these components and together, this should be glutathione, decreases with age in red blood cells. Red blood cells readily take up AKG, driving glutathione production. Red blood cells are particularly sensitive to lack of glutathione because remember, they're carrying millions of oxygen molecules around with hemoglobin, and so they're sensitive to reactive oxygen damage, and even hemoglobin is sensitive. And so what we think is one particular protective mechanism of alpha-ketoglutarate with age is that it's restoring glutathione, increasing red blood cell function, and probably assisting oxygen delivery to muscle, which can affect both exercise and probably help people as they get older and start to have reduced muscle function. So we're very interested in this pathway right now and looking at it in some detail. I should point out that increasing oxygen delivery to peripheral tissues might be very beneficial in the context of COVID-19 as well, in patients that are suffering from acute respiratory distress syndrome. So we've given some thought to whether it might be useful acutely to help people get through that very late stages of the virus.
It's also interesting in the context of COVID-19 because reduced oxygen delivery is one of the major risk factors for major predictors of survival in affected COVID-19 patients. So what happens is their pulse ox goes down dramatically as they get sick. And AKG, if it's improving oxygen carrying capacity, which we think it is, may be one way to get people through this very difficult, challenging time, allow them to survive until their system works better to clear the virus. So we're interested in it from that perspective as well. And I think that one thing I want to mention is that learning about aging is useful for not only chronic disease, but for these viral infections too. So one of the theories about aging has always been that nitric oxide levels go down with age, and that improving nitric oxide may be a way of slowing the aging process. And so this is nitric oxide production with age, and this is the decline in production again in 2 different studies. So you can see a really dramatic effect. It turns out most people think inducible nitric oxide synthase is the big driver of nitric oxide production, but in endothelial cells, there's also an endothelial nitric oxide synthase. And when you're under inflammatory conditions, that activates this enzyme to produce nitric oxide, and that serves as a primary defense mechanism in innate immunity when you get a viral infection. And we know that endothelial cells are vascular endothelial cells are heavily influenced by this virus, and maybe one site where severe complications are arising. And so it's been shown a long time ago with another virus that if you just increase nitric oxide levels, you can improve the resistance to this is influenza, improve resistance to influenza. So we were curious about this and wrote a concept paper on this recently. And it was the best review we've ever gotten because this was not really our field. And the reviewer came back and said, you know, there are a lot of polymorphisms in endothelial nitric oxide synthase. Maybe you should have a look at them and see if it correlates to anything. And so what I plotted here, there are many alleles of nitric oxide synthase. I won't go into it. What we're looking here is the prevalence of the wild type or the functional allele of eNOS. A lot of people have mutations which reduce function, and the death per million due to coronavirus. And there are a lot of complicating factors here causing mortality due to coronavirus. How how good the treatment is in different countries, and a whole range of other things. But the striking thing here is that the countries, at least at this time when we did this study, that had a relatively low mortality were also the ones where you have the highest prevalence of the wild type allele of eNOS. So again, we think using aging, we're learning about interesting things regarding the response to acute infections. And of course, there are ways to increase eNOS. And a lot of these molecules have been linked to aging. Statins have been linked to aging. Resveratrol has been linked to aging, as well as adding back hormones like estrogen. And I won't mention blue pills, but that's another way of increasing nitric oxide synthase. And finally, vitamin D, which has been shown to slow aging in animal models, is also a regulator of eNOS. So we're looking heavily at this pathway, both with respect to aging and viral response. Coming back to eNOS, what we've done is set up a healthy longevity program. We have about 40 faculty in it now, some of which are like me working on basic biology of aging, but we also have a focus on muscle aging, brain aging, vascular aging, and we have a new group we're adding on regenerative medicine and aging, and another group looking at female reproductive aging now. So we're really trying to expand this group. We're very collaborative, and we're happy to work with anyone who has ideas on how to test the aging process. But this is all fine. So what I've convinced you so far is if you're a mouse, we got you. We can make you live longer, healthier, but most of you listening probably aren't mice. And so how do we get human? So I think we have three goals. One is to validate that interventions work in humans. The second one is to optimize and personalize those interventions. I don't think any single intervention is going to work in everyone. We have to understand who responds to which kinds of interventions. Even exercise, which is pretty much clear, works to extend lifespan. Even that is probably individual specific. The right type of exercise for each person is probably different. And then we have to do things in ways that scale. So while I work with companies on some exotic things like, stem cell injections and stuff like that, we're not in the academic side working on $100,000 interventions right now. What we're trying to do is look at relatively cheap interventions that might affect aging that we can rapidly move to the population. So how do you validate a clinical trial for intervention that affects aging? So pharmaceutical companies have taken sort of the first approach. And that, again, relates to the fact that aging is not a disease and they have to get reimbursed, so they need a disease. So they may get interested in some pathway or drug because it targets aging, but then they rapidly switch to doing clinical trials trying to treat disease. And so far, this has not worked so well, and it's led to several high-profile failures. I think that, while slowing aging may be very effective while you're still healthy and that network is still functioning, whether it works after you're in a state of chronic disease is an entirely different question. And so I think that I understand why people do these studies. Companies I work with do them, but I'm a little bit concerned that this is not the best way to get to healthy aging. Second approach you can take is to prevent disease. And there's a large study called the TAME trial with metformin going on in the US right now, 3,000 people, 14 clinical sites. Study goes on for 3 years. And the idea is that metformin, which is a diabetes drug, well, since it slows aging, it won't just work to prevent diabetes, it'll also prevent a whole range of other diseases of aging. And that's the endpoint that they're going after in this clinical trial. I don't have any problem with this, except that it's extremely slow and expensive. And I don't think any of us know which interventions are going to work in humans right now. So I don't want to spend $60 million on one test. So what we're doing here is smaller, more targeted, and something that we can compare agnostically different kinds of interventions. So what we're using are aging biomarkers. And so I want to spend a little bit of time talking about what aging biomarkers are. Just to give you an overview for the clinical study on AKG, the top one is sponsored by the company in the United States and is ongoing right now. The bottom one is something we have planned at eNOS, and they're very similar, so I'll just go through them briefly. The idea is to take 45 to 65-year-olds. We don't want to start these studies in older, frail people, again, because I don't know whether things will work after you're in that condition or not. They may. But what we want to do is take people that are still largely healthy but at risk of disease. And those we think are the best place to start to look at interventions in slow aging. Now, you might argue that the best place to start is as soon as you become an adult at 20, and that may be true, but it's a lot harder to think about an intervention that you give to a 20-year-old than for 50 years. So we want to start first in this middle age range. Interventions are 6 to 9 months, and we want to look at aging biomarkers and a range of other things. There's a lot more things we're looking at in the Singapore study. And so what we're trying to do really here is to build an iterative testing strategy. So we've refined how we do mouse experiments now to make them as close to human studies as possible. Shorter term mouse studies using aging biomarkers and frailty in mice as well as in the human studies. Therefore, we can find things that work in animals, move them rapidly to humans, figure out what works, go back, try to refine things in mice, or even combine different interventions together and keep working back and forth. We're also interested in mechanisms. We have a few disease models we're intrigued by, and we're working with Dean Ho to try to do precision dosing. So instead of giving all the animals one dose of a supplement or drug, let's figure out how much each animal needs and optimize per animal. And if we can get that to work, I think the effects might be much larger, and we'll move that into human studies as quickly as possible.